All right, we ended the last slideshow presentation here. Cross-bridge cycling. Remember, you can review all of that, all of the steps in cross-bridge cycling, but the effect of this is you are moving one myofilament, myofibril, a short distance. That's what you're getting. You're getting one myofibril or one muscle fiber. Let's say it that way. One muscle fiber to contract. And one muscle fiber isn't going to give you much of a contraction. You need more than one muscle fiber. So what does that mean? That means that we got to move on from here. We have to, we'll get back to the threshold stimulus in a moment. What you need in order to get a combination of muscle fibers together is to recruit what's known as a motor unit. The motor unit is exactly what it says here, a motor neuron. So a motor neuron is the nerve that goes to muscle. That's called a motor neuron because the effect of muscle is to create a motor. The motor is the muscle, right? So we call those motor neurons. Well, what a motor neuron does, and you can see, let me get my cool little pointer, my laser pointer here. Watch this. Love this thing. All right, so if I have that motor neuron, one motor neuron. Yes, the one motor neuron comes down here, and this is the axon, and these are covered with Schwann cells, and that's a myelin sheath covering. We're going to get to that in the next lecture. But here's this motor neuron coming down. Yes, it splits apart, and it goes to multiple muscle cells. So this one motor neuron innervates a few muscle cells. It could innervate 2 or 3 or 5 or 10, or it could innervate thousands or tens of thousands of muscle fibers. The effect is the same. As this one motor neuron sends a signal to all of the muscle fibers that it is innervating, all of them will contract. So one motor neuron plus all of the muscle fibers it controls. When that one motor neuron fires, it will fire all of the muscle fibers that it is connected to. That, as I say, it could be a few, it could be tens of thousands. So you could get a little tiny bit of power out of it, you could get a huge amount of power out of it. But if you need to add more power, you need more muscle fibers contracting at the same time. How do you do that? You add another motor unit. So you have motor unit number 1, and this one they've color-coded it, so the muscle cells that it is innervating are purple. So here's a purple one, here's a purple one, here's a purple one, right? You can see all the purple ones. So that is called a motor unit. So in this case, we call it motor unit number 1. There's all the purple ones. The second one is motor unit number 2. Motor unit number 2 has all the red ones. All right? So if you need more power, you add motor units. You add them sequentially. Right? So the whole muscle consists of many motor units. Coarse movements are produced with large numbers of fibers in a motor unit. That means that one nerve will control tens of thousands of muscle fibers, like your quadricep muscle in the upper part of your thigh. One motor unit will consist of tens of thousands of muscle fibers. Precise movements are produced with fewer muscle fibers. Your fingertips, your eyes, the movement of your eyes, the muscles that move your eyes around are very finely controlled. And so one motor neuron might control just a few muscle cells. You need very precise. So you would still add motor units, but you would add another motor unit that has maybe 10 or 20 or 30 more muscle fibers, and then another and another, depending on how you need to move your eyes around. You need very fine control. So let's make sure you understand that precise movements will still have one motor nerve, but that motor neuron controls maybe 10 or 20, just a few, a small number of muscle fibers. Whereas if you need strength, you need power, you're going to have one motor neuron controls thousands or tens of thousands or even more muscle fibers at the same time. So you need power. The motor units are going to be very large numbers of muscle fibers. Precise or tiny or very, very controlled movements need motor neurons with very few numbers of muscle fibers. So we would say that precise movements are produced with fewer muscle fibers in a motor unit. All right. Now, so you got to combine these motor units together in order to get sustained contractions. Well, of course, now you know how a muscle fiber contracts, right? You send calcium, you get the calcium onto the troponin, and then the troponin moves the tropomyosin, and then myosin and actin bind, and the myosin moves, the power stroke moves, and that pulls the actin a little ways. And you have millions and millions and millions of these interactions happening, and you're moving that one sarcomere. Well, you need a whole bunch of those. So you need, I would say, trillions. You need trillions of these sliding filaments to slide in order to move anything. Then you have to combine them together. So you combine them together into motor units, as I said in the last slide. Smaller motor units, so that means smaller motor units have very few muscle fibers attached to them. And they also have another phenomena. The nerves themselves are smaller diameter axons. And we're going to learn that a smaller diameter axon sends signals a little bit more slowly, just a little bit, not a lot, but a little bit more slowly, and in a slightly more controlled fashion. So when you have to pick something up, those are recruited first. At first you don't know how heavy or how delicate the thing is you're going to pick up if you're not familiar with it. So you're going to pick up a Cheerio, one little Cheerio, the cereal Cheerio. You're going to pick it up. You're going to recruit a small number of motor units with small diameter axons to recruit first. You're not really sure what you need. Then it turns out you were fooled. That was a Cheerio made of lead. I don't know, that's silly, but it's heavier than you thought. So now you're going to need more motor units. Now you're going to start to recruit larger motor units. That means larger motor units means more muscle fibers with larger diameter axons. So they're going to get a faster signal and a stronger signal, and they're recruited later on. So you only recruit them, you recruit what you need to recruit in order to get the job done. Okay? But you know, inside any muscle cell, any skeletal muscle cell for sure, that these myosin and actin groups are binding and unbinding and binding and unbinding and binding and unbinding. And they're going fast through this cycling, through this cross-bridge cycling that you learned about. Well, you need to add these things together into trillions and trillions of interactions. And so the process by which that happens is what's known as summation. Summation, recruitment you already learned about. Recruiting motor units is how you can recruit more muscle fibers to get in on the action. But summation is the process by which all of these muscle fibers help each other out. Right? One contracts and another one has to relax because it's got to get more energy or more ATP or more calcium or whatever it's got to do. And so it detaches from the actin, and then another one attaches, and so back and forth and back and forth. These things help each other out. So they summate their interaction patterns. And recruitment, you recruit a number of motor units that you need to perform a task, and they produce sustained contractions of increasing strength. Right? What does that mean for you? That means you don't notice all of these cross bridges attaching and detaching. What you notice is a contraction seems to be a smooth movement to you. Right? That's how you interpret it. You pick something up and it doesn't seem like everything's herky-jerky inside your arm where these myosins and actins are attaching and detaching and attaching and detaching. That happens so many times a second, so fast, that you don't notice it. What you notice is you say, oh, this is a smooth movement. My muscles move in a very smooth fashion. The other thing about muscles is there's this underlying idea called muscle tone, or what we call tonus. Muscle tone, it means that your muscles, most of your muscles in your body, are in some state of partial contraction. Even if they're resting, you say, oh, I'm just resting. Well, those muscles they're not completely, like if you were anesthetized, then you could say, yes, my muscles cannot contract, they do not contract, they are completely at rest. But at any other time, anytime you're conscious for sure, those muscles are in some partial state of contraction. and they're ready to contract even more. So we call that muscle tone. So muscle tone is the sort of baseline tightness or tension or contracted state that all of your muscles are in at any given time. And of course your muscle tone will change depending on your situation. If you would say you're at rest, you're sitting in a chair, you would say, well, I'm just resting. I don't have any muscle tone. Well, of course you do. You have some muscle tone, some degree of muscle tone. Then you stand up. You need more muscle tone in certain muscles to hold you up. We would call those postural muscles. All right. So if you're going to recruit more motor units, that means you have to do something that requires more effort. Okay. So recruitment, increasing the number of motor units activated to produce more force. That's the whole point. If you have one motor unit contracted, you get a tiny little bit of force out of it. If you have 2, 3, 10, 20, 1,000, if you start to recruit more and more and more motor units, you are going to produce more force. Simple as that. Certain motor units are activated first. Others are activated only when the intensity of the stimulus increases. So, I mean, if you're lifting weights and you're trying to find your 1 repetition maximum, that's called the 1 rep max, you're going to recruit as many motor units as you possibly can to finish that task. Otherwise, most of the time, you're recruiting only enough motor units to do whatever you need to do. Right? As the intensity of the stimulation increases, recruitment of motor units continues until all motor units are activated. If you activate all of the motor units, that's as strong as you can be at that particular time. Right, so a muscle contraction can be observed by removing a single muscle fiber, a skeletal muscle fiber we're talking about here, and connecting it to a device that senses and records the changes in overall muscle length of the fiber. Right, so that can be done. There are experiments to do that. I'm going to show you a picture in a moment. Now, in this case, in the picture that I'm going to show you, there's no nerves attached to it that are telling the muscle to contract. They've connected an electrical stimulator. So this machine produces voltage, right? And they can vary how much voltage that they produce, and then they can measure how hard that thing contracts. So let's look at this thing. All right, so here it is. This is straight out of your book, by the way. Okay? We're going to go to, I'm going to change things a little bit. Okay, so you see here's a muscle. So they took this muscle out of now normally the typical thing is to take a muscle out of a frog leg. But we didn't do that here, okay? We'll just say here's a muscle, and it's attached at both ends. So it's attached up here, and it's attached down there to this device. And then, instead of a nerve, they got this electrical wire, and the electrical wire goes to this voltage generator, right? Think of a battery. So it's ready to produce any necessary voltage that's needed to get this thing to contract. Now the thing is too, if I give this thing a jolt, a shock, and nothing happens, that was not enough stimulus to get the muscle to contract. So now we go back to this slide that I passed by. This thing, threshold stimulus, okay? Threshold stimulus. You must have enough stimulus in a muscle to get it to fire. If you don't have enough stimulus, it won't fire. And so the muscle is waiting to get enough of a signal to contract. When it does, when the strength of the stimulus reaches threshold, an action potential is generated. Okay? That's going to be true for nerves too. We're going to get to that when we talk about nerves. But this is the precursor to nerves, right? When you reach threshold, you're going to get an action potential. An action potential is measurable. Reaching threshold in the case of a muscle means you have to have enough acetylcholine cross the synapse to attach to the acetylcholine receptor on the muscle cell membrane. When you have enough of that, then you're going to generate an action potential in the sarcolemma. You will get a muscle contraction when that happens. You will, every time. So when the impulse spreads through the muscle fiber, calcium is released, sarcoplasmic reticulum, you get cross-bridge formation. That, by definition, is muscle contraction. So here's the take-home, right? Right here. We're going to star that. The take-home, 1 action potential from a motor neuron releases enough acetylcholine to produce a threshold stimulus in a muscle fiber. What does that mean? It causes a muscle impulse. The muscle impulse results in a muscle contraction. So one action potential from a motor neuron releases enough acetylcholine to produce the threshold stimulus in a muscle fiber. And what do we say? Threshold stimulus. When the strength of the stimulus reaches threshold stimulus, an action potential is generated through the sarcolemma, calcium is released, you get a contraction. But you have to reach threshold. Now we go back to our experiment here, okay? So what they're doing is they're initiating a stimulus, okay? And so we know, they know this experimental situation is set up so that they know how much stimulus they need to inject, how much voltage do they need to send to this muscle to get it to contract. Okay, let's say they do that, right? They push the button, zap, they're sending a signal to the muscle. It enters the muscle. When did we do that? Right here. Right there. That is when they introduce the stimulus. And then you say, yeah, okay, where's my contraction? What happened here? Well, what happens is right here, this is what's known as the latent period. Okay, the latent period looks like on a graph, it looks like, yeah, I get nothing. I'm getting no contraction. See, this thing over here, muscle tension. It's telling me there is no muscle tension. What do you mean? I haven't changed it from the baseline. How can there not be a muscle contraction for this length of time? This latent period, what's happening there is you already know, the excitation, contraction, coupling processes. All of those processes, every single one of them are occurring in this interval of time. Okay, from here, I know I'm not drawing a very straight line there. All right. Between those two arrows, that's the latent period. The latent period, the muscle itself is not contracting. But what is happening? Every process in excitation, contraction, coupling. This takes 0.0, about 0.02 seconds. What is that? Oh, this is hard to draw words with. Yeah, that's pretty bad. Or we really say, this is how we really say it, 10 to 20. This is average too, okay? This looks terrible. 10 to 20 milliseconds. So that's how you'll see it sometimes written out. MSEC. What does that mean? 10 to 20 milliseconds. That's the same thing here. 0.02 seconds is the same thing as 20 milliseconds. So it takes about 10 to 20 milliseconds. That means for 10 to 20 milliseconds, you're just waiting around and nothing's happening. Well, all that other stuff is happening. Right, the sarcolemma is depolarizing. The T-tubules are depolarizing. The sarcoplasmic reticulum is letting go of calcium. The calcium is binding to the troponin. The tropomyosin is moving out of the way. The myosin is binding with the actin. All of that stuff, all of those things are happening right here. All of that stuff. But of course, there's no force generated. And then, whammo, you get force generation, okay? Force generation, contraction period. All of a sudden, myosin is pulling on actin. You get your classic muscle contraction, right? And then it all ends. If there's no more stimulation continuing to stimulate the sarcolemma and release all that calcium, just as we talked about, if that's done, and in this particular experiment, if we are not continuously producing voltage from this thing to contract that muscle, it relaxes, right? So you can get the relaxation period. Muscle tension disappears, back down to normal, back down to baseline tension. That's it. This process, this one process right here in front of you is called a twitch. This is a twitch. Right? So here it is in another slide. Same thing. Latent period. You get a time of stimulation. You get a hesitation to this point. Then this thing, then you get the period of a muscle contraction. Then you get the period of muscle relaxation. Same thing, 10 to 20 milliseconds in there. Relaxes, twitch. This is called a twitch. See that? Twitch, 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 twitch. Contractile response of a single muscle fiber to a single impulse. That's a twitch. boom, done. It's still a muscle contraction, but you can't really do anything with a twitch. But a twitch is necessary. So you got to have one twitch. Now we have to combine twitches. So I put this slide in here as an extra in this slideshow that I have to remind you of all the stuff that's happening in that latent period, right? Everything that we went through in that one video, a couple of videos ago, took me 10 minutes to go through this whole process, right? That happens in 10 to 20 milliseconds in normal skeletal muscle. So here it is, right? So here's a twitch. What they're doing here is they're stimulating the muscle at these arrows. Stimulate, then let it rest. Stimulate, let it rest. Stimulate, let it rest. Stimulate, let it rest. We're not stimulating it fast enough to get these twitches to combine together. When they combine together, that's called summation. So it's summating twitches, okay? So if the fiber twitches once, you get a twitch. If you start to combine them together, what that requires is you have to you have to give the stimulus at shorter intervals. So here's a twitch, but then I give this second twitch here, this twitch. I gave that twitch before it had a chance to relax completely, right? And now, whoop, the tension went up a little bit. And then I didn't let it relax completely again. Whoop, the tension goes up again, right? So I can keep doing that. I can keep combining these steps. So this is called the staircase phenomena, or what's known as treppy. And you'll see that in the next slide. Treppy. Oh boy, this is awful to draw words with. Wow, that is a weird looking P. Anyway, treppy. Treppy is German for stairs. Right, it looks like stairs. Stair, step, stair, step, stair, step. You keep combining twitches until you get a sustained contraction. From this point on, you get a sustained contraction. Okay? So now let's go to the next slide. So these are called, produces sustained contractions. It leads to partial or complete tetanic contractions. Not titanic, tetanic. Tetany. This is called tetany. This is straight out of your book. This is a graph out of your book. This is a twitch, as I told you already. Twitch, let it relax. Twitch, let it relax. Twitch, let it relax. You're not getting anything. You're not getting any movement or any particular useful activity out of that muscle. So there, less than 10 stimuli per second. We'll have to stimulate it faster than that. That means the nerve that is stimulating your muscles is stimulating faster than 10 times a second. 20 times to 50 times per second. Whew, that's a lot. So your nerves are bam, 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 telling your muscles contract, 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 and you get wave summation. So this is treppy, right? There's a step, there's a step, there's a step, there's a step, there's a step. All of these things are summating. This is called wave summation. As those waves of contractions summate, add together, you get into a state that's known as incomplete tetany. You're still getting it. It's contracting and relaxing, contracting and relaxing. Now you can only measure this on, in these special kinds of equipments. You can't measure it within yourself. You don't feel this. But what you're waiting for is tetany. Tetany is the ability to combine all of these, to wave summate all of these muscle fibers into what feels to you as a continuous contraction. That's called tetany. Right? So you hold a mug of coffee in your hand in front of you, and as long as you hold it there, your muscles are in tetany to help you to hold that thing there. What we know is you cannot do that forever. At some point, your arm's going to get tired, right? And when that happens, then we get into what's known as muscular fatigue. So we got to talk about this phenomena of fatigue. So all these motor units, all these muscle fibers, they're all churning away, and eventually they just can't keep doing that. And so they get fatigued. That's called neuromuscular fatigue. And so let's talk about that. Now, when you're going to relax, the first thing is when you relax a muscle, remember the acetylcholine comes out of the end of the nerve, and it binds with its receptor, okay, on the muscle membrane. as long as that acetylcholine is intact, as long as it's still there in the synapse, it will continue to bind to its receptor, and it will continue to create a depolarization at that spot. You can't keep doing that forever. You have to stop that. How are you going to stop it? Well, one of the ways to stop it is this. Acetylcholine esterase. So this thing ACE, and we've talked about that before, an ACE. An ACE is an enzyme. An enzyme, in this particular case, is going to break down acetylcholine. So acetylcholine esterase sits there in the synapse waiting for acetylcholine. Once it sees it, it decomposes acetylcholine remaining in the synapse. No acetylcholine, no binding to its receptor, no depolarization of the membrane, no contraction. Okay? Muscle impulse stops when acetylcholine is decomposed. Of course. Makes sense. Stimulus to the sarcolemma and the muscle fiber membrane ceases. No more action potentials. The calcium pump, there's a calcium pump in the sarcoplasmic reticulum, okay? It's in the sarcoplasmic reticulum. Calcium pump grabs onto all the calcium that's sitting around the myofibrils and pulls it back into the sarcoplasmic reticulum. So now it's storing it up again. It's waiting. Okay? And the troponin and the tropomyosin complex covers the binding sites on actin. Myosin and actin binding are now prevented. Muscle fiber relaxes. That's how relaxation occurs. You got to have energy for this. Remember, yes, I'm going to go back here. You have to have calcium, of course. Yes. We all know about the calcium. Calcium's not enough. You got to have, what else you have to have? You have to have ATP. Okay? So when ATP and calcium are present, you're going to get a contraction. You have a small amount. What do we say a small amount? What is a small amount? A small amount means that you have enough ATP sitting inside your muscle cells for about 5 to 6 seconds of full out contraction. So if you were to say run from that bear, remember it's always a bear, you're always running from a bear. If you're running from the bear, you got 5 to 6 seconds at full speed. And then you're going to start slowing down because you've run out of ATP. Okay, how are you going to get more ATP? If you're going to generate ATP very, very fast, there's a way to do it inside your cells, inside a muscle. Muscles have this stuff called creatine phosphate. Creatine phosphate will give up its phosphate to the ADP and recreate ATP. Okay, so here's how it works. When cellular ATP is high, that means when you're at rest. When you don't need the ATP, you make ATP and you store some up. Not a ton, but some. You store some up, right? So what that means is you don't need more ATP. So the excess ATP, so we're going to consider this to be excess ATP right there. That excess ATP gives up its phosphate group to creatine. Creatine is kind of like a storage molecule for phosphate inside a cell. All right, so the creatine hangs onto that high energy phosphate for use in a rainy day. Now the bear comes along. Here's the bear. Okay? Bear. Yeah, this is not the best way to draw letters, I'm telling you. Bear. There. The bear. Bear comes along. ATP is low because you're running, right? You're running from the bear, so your ATP is drained. You need more ATP. How are you going to get it? Creatine phosphate is your little storehouse of phosphate. It gives the phosphate to ADP and then recombines it to make ATP. Now ATP can go over to the myosin and actin complex and disassociate it, right? But you can't do that for long. ATP reserves 5 to 6 seconds and then you're out of ATP. Creatine phosphate can rekindle that ATP for about another maybe 10 to 15 seconds. Not a long time, but you got a little bit more time. So you can still create ATP. You can still go at a pretty fair pace for a fair amount of time. So you put these two together, 5 to 6 seconds with number 1, 5 to 6 seconds there. Number 2, you're going to get another 10 to 15 seconds. So maybe you got 20 seconds, 15 to 20 seconds total of top speed muscle contraction with the ATP that you already have raring to go, or the creatine phosphate, right? After that, you've got to depend on cellular respiration. Now you have to break down glucose. We go back to like the first or second week of class where you have to break down glucose to get ATP. All right, so the first thing is you're going to do it anaerobically. Remember, that's called glycolysis. Glycolysis is anaerobic respiration. The anaerobic part, glycolysis, occurs in the cytoplasm, produces little ATP. Remember, you only get yep, there it is right there. You get 2 ATP. All right, well, you got 2. That's more than you had before because you were out. You were out of ATP. So glycolysis will let you go for a little bit longer anaerobically. That just means you can't get enough oxygen to those cells to generate the ATP that you would like to have. Okay? So that can go on for another maybe 30 seconds, maybe a minute. But then the anaerobic glycolysis, the anaerobic phase, is going to also run out of ATP. The only way you're going to get enough ATP is this, the aerobic respiration, the aerobic phase of cellular respiration. Remember citric acid cycle, electron transport chain, that's where you're going to get all of this ATP. Now that happens in the mitochondria. That's why muscles have tons and tons of mitochondria. Now, there's another source. So this is the source to get energy, to make ATP. But remember, ATP, you're only going to get the most ATP if you have oxygen present. Well, I just told you, if you're running from a bear, you've gone through anaerobics, you don't have a lot of oxygen left over. There's this molecule called myoglobin that is specific to muscles. Muscles have, and you can tell that, myo means muscle, right? So myoglobin is a source of oxygen when oxygen is running low in a muscle cell. And that oxygen will allow you to continue in the aerobic phase of cellular respiration to make more ATP. So myoglobin allows you to keep going for a little bit longer. So as you're cranking along, as you're running from the bear, you've used up your the immediate source of ATP. You've used up your creatine phosphate to make more ATP. You've gone through the anaerobic phase of cellular respiration. You're into the aerobic phase of cellular respiration. And you are using up all of the ATP that you had. And remember, most of it was made through aerobic respiration while you were at rest, while you didn't need the ATP, right? It's just waiting around. You incur what we call an oxygen, where's my little, there it is. You incur an oxygen debt. An oxygen debt is the same thing as if you spent more money than you have. You're in debt. You don't have more money. An oxygen debt means I need to rekindle my storehouse of energy in the case of your cells. Or if you need to rekindle the storehouse of money in your bank account, for example, you have to repay the money. So oxygen debt is money, is oxygen that you were loaned, or energy that you were loaned that you have to repay at a later time. And so we refer to that as an oxygen debt. Okay? So it's during rest or moderate exercise, respiratory and cardiovascular systems supply enough oxygen to support aerobic respiration. Right? That's rest or moderate exercise. We're not talking about rest or moderate exercise. We're talking about beyond that. So when you run out of oxygen, remember glycolysis makes pyruvate normally, and pyruvate ends up getting changed, but it goes into the mitochondria, and then you go through the tricarboxylic acid cycle, Krebs cycle, and you make more ATP. Well, if you don't have enough oxygen around, there is a shift in metabolism. Right? So again, you're running from the bear. You're running and running and running. There's a shift in metabolism from aerobic to anaerobic. In anaerobic, the pyruvate now is turned into lactic acid. So you're familiar with lactic acid, right? The feeling, the burning sensation in your muscles where they can't keep going. You have to pay off the oxygen debt. So what is the oxygen debt? That is the amount of oxygen needed by liver cells to convert the accumulated lactic acid to glucose. So what does that mean? It's telling you right here. The blood is full of lactic acid. It has to be converted back to glucose in order to restore muscle ATP. That's the oxygen debt. How much oxygen do you need in the liver to convert lactic acid to glucose? Remember, glucose will then go through the whole cellular respiration, and it will restore the ATP that you had to use during the time the bear was chasing you. That's called an oxygen debt. You have to pay the debt. How do you pay the debt? You have to stop running fast. You have to sit down. You have to breathe real fast. You have to get enough oxygen in there. Eventually your breathing rate will slow down. Your metabolism slows down. You have paid your debt. That's how you pay off the oxygen debt. Here it is. Right? So you started off with glycogen in the liver. That's where it is. Glycogen is converted to glucose. Glucose is metabolized to make ATP. And normally, you get pyruvic acid out of the deal. Oops, I don't have enough oxygen, so now I'm going to create lactic acid in an anaerobic situation. That's the burning sensation in your muscles. All right, you have to pay the debt. Paying the debt means you have to use energy from ATP at rest in order to create more glucose in order to restore your glycogen stores. Round and round and round and round and round that goes. So when you get to the point when you can't contract your muscles, when you cannot run as fast as you need to, when you're running away from the bear, it's always a bear, I know, I keep saying that. That's called muscle fatigue or muscular fatigue. What is that? That's the inability to contract muscle, or at least the inability to contract muscle at the same pace, at the same rate that you were contracting it. You just can't keep going. Take a heavy object, a book, a big book, take your computer, take a chair, I don't care, take anything and stand up and hold it in your arms straight out in front of you. See how long you can hold it up there. You'll go into muscular fatigue. Muscular fatigue will tell you you won't be able to hold that thing up anymore. It's the inability to contract that muscle any further. Right? There are a lot of reasons. Decreased blood flow is one. Ion imbalances across the sarcolemma. Loss of desire to continue because lactic acid builds up. Lactic acid starts to give you this acidy, burny feeling, and that's very unpleasant. And that, now, it's a bit controversial. It's not so much the lactic acid that's the problem. It's not so much the lactic part of it. It's the acid part that's really the problem. And your enzymes don't work very well when the pH changes, if you remember way back when we talked about that. If you keep doing that, you may end up into what's called a muscle cramp. A muscle cramp is a sustained, involuntary muscle contraction. That's the important thing here. You can contract your muscles really, really hard, but that's a voluntary. This is involuntary. You're not trying to contract your muscles. They're cramping. You don't like it. It feels awful. Now, there's a ton of explanations, or what we refer to as hypotheses about muscle cramps. There's no one reason why people cramp muscles. All it is a muscle cramp is a sustained involuntary muscle contraction. So bits and pieces here. One of the things to remember about muscle metabolism, or the cellular metabolism that goes on with contracting muscles, is heat is a byproduct. Now it turns out, as it says down here, more than half of the energy released in cellular respiration becomes heat. More than half. Less than half is transferred to ATP. So that means if you're moving around, you are using ATP. You have to create ATP, right? More than half of the energy that goes into making ATP actually goes into the production of heat. That's what makes you a homeotherm. Homeotherms maintain a constant body temperature. One of the ways you do that is that you move around. By moving, you make heat, but you're also making energy. So muscle cells are a major source of body heat, right? So I've said here, if you need to warm up, if you're cold, get up, move around, right? That's a voluntary way to make yourself warmer. Or if you don't like that idea, don't worry. your body has a solution to that. It's called shivering. You will not be able to control your shivering. Why? Your body is using the production of energy to your muscles to make you wiggle around uncontrollably. We call it a shiver. And that generates body heat. You're going to warm yourself up at least a little bit by shivering. And then you make all that heat, and the blood transports the heat throughout the body core, keeping all your internal organs alive. Before stimulation determines the amount of force it can develop. What does that mean? The optimum starting length is the resting length of the muscle fiber. Resting length, okay? So this is resting length. That means it's not contracted and it's not extended. It's in the middle of its movement. A stretched muscle develops less force. You can prove it to yourself. Straighten your arm and try to lift something heavy. Or have, try to bend your arm when you have something very heavy in your hand. If you try to hold it, if you try to bend your arm and it's very heavy, it's very, very hard to get your elbow to bend at that point. If you put that heavy object in your already bended elbow, you're stronger. So that means out here you're overly stretched. That means your elbow is just about straight. That means you have less overlap of the myosin head groups with the actin filaments. You're weaker. Over here, on the other end of it, when it's overly shortened, that means you have your elbow bent as far as you can bend it. Well, you can't bend it anymore, so you can see there where the actin filaments are running into each other. There's no more room. There's nowhere else to bend. You can't develop any more strength. That's it. That's as strong as you can get. So you're strongest when your actin and myosin are considered to be optimally overlapped. Right, so that's called a length-tension relationship. The length of the sarcomere or the length of the muscle fiber is dependent or will determine how much tension you can develop in that muscle fiber. And that's a relationship, the length-tension relationship. There are various kinds of muscle contractions. You're very familiar with isometric. If you put a book or a coffee mug in your hand or something in your hand and hold it there and don't move your arm, your muscles are in a state of isometric contraction. And remember, iso means the same, iso, and metric means length, right? So an isometric contraction is one where the muscle stays the same length, does not change length. Okay? So the muscle stays the same length. But if I'm going to move my muscles, which are going to move joints and move bones around, there's two different ways that I can do that. One is concentric. A concentric is a muscle shortening. We just talked all about muscle shortening. Sarcomeres pull in together, muscle fibers shorten and contract, you bend your elbow. But at some point, you have to straighten your elbow. But you don't just let your muscles flop straight, right? You slowly allow them to go back to the starting position, right? So when you do that, that's called an eccentric contraction. Now it's a little weird, but what you're really doing there is you are allowing or slowly lengthening the sarcomere back to its original length. So when you concentrically contract the muscle, it shortens the sarcomere or shortens the muscle fiber. Of course, we've been through all that. But at some point you have to allow that thing to lengthen back to its normal length. And that still involves the myosin head groups grabbing onto actin. But instead of pulling the actin toward the middle of the sarcomere, it's slowly letting it lengthen back to its normal length. So that's known as an eccentric contraction. So these three types of contractions are the most common that you're going to see in skeletal muscles. If it's isotonic, that means the length of the muscle is changing, right? Changes length. So you can have two types there, concentric or eccentric. The other kind of muscle contraction is isometric. No change in the length of the muscle fiber.